Okay, my name is Yanbin Yin. I am an associate professor of the University of Nebraska-Lincoln. I want to first thank the organizers for the opportunity to present. Today I will talk about eCAMI, the carbohydrate enzyme annotation tool that we recently developed. First, I want to acknowledge participants of this work. Jing Xu is a master student of Nankai University in China. She was the main developer of the tool under the supervision of Dr. Han Jiang. Jing Wang and Philip are two members of my lab at UNL who were also involved in this project. This work was supported by an ASF grant. So this is the outline. I will first introduce carbohydrate active enzyme or enzymes and their annotation. And then I will talk about eCAMI algorithm and performance evaluation. And last, I will talk about the application to enzyme EC prediction using eCAMI. So as the name indicated, carbohydrate active enzyme or enzymes are enzyme targeting on carbohydrates, specifically glycosidic bonds found in various polysaccharides or oligosaccharides. This chemical linkages form between two monosugars such as glucose via alpha 1,4 linkage will give you maltose. Similarly, glucose-galactose via beta 1,4 linkage will give you lactose, and glucose-fructose via alpha 1,2 linkage will give you sucrose. There are probably over 30 different monosugars found in nature. They can be connected through a huge number of possible linkages to form polymers of polysaccharides with different lengths. This makes carbohydrates the most structurally diverse molecules on Earth, as each glycosidic linkage requires a different enzyme to build or break. The sequence diversity of enzymes are extremely high. Since 1998, a database called KISI started to collect experimentally characterized enzymes and classify them into 6 classes based on their molecular mechanisms. And these 6 classes are further classified into 400 protein families according to sequence similarity. Among the 6 classes, glycoside transferases, or GTs, are used to synthesize carbohydrate polymers by build glycosidic bonds. So GTs include 110 protein families so far. Other classes like GH, glycoside hydrolysis, PLs, polysaccharide lysis, and CEs, carbohydrate esterases, are used to degrade carbohydrates, and AAs, auxiliary activities enzymes, are enzymes targeting lignins. Lastly, CBMs, carbohydrate binding modules, often coexist with different catalytic domains to form multi-domain proteins. So KISI database appeared twice in NR's database special issue, which together has been cited almost 8,000 times. In addition to experimentally verified enzymes collected from literature, KISI also perform sequence homology search, such as blast search and sequence motif analysis, to populate each of the 435 families. Together, the KISI database contains over 1.3 million proteins today, but only 2% of them are experimentally studied. Given their functions in carbohydrate metabolism, we will imagine to find more of these enzymes in carbohydrate-rich environments. Indeed, some human gut bacteria dedicate up to 10% of their genes, including enzyme, because they feed on the fibers in our diets that is indigestible by humans. In addition, enzymes are also critically important for bioenergy and biomaterial production. Other application includes plant disease study, microbiome of plants, human cow rumen, and also soil for global carbon recycling research. With the KZ database given a newly sequenced genome, one can perform blast search to annotate the genome for enzymes. In 2012, we developed a database called DBCAN, which contains a signature domain hidden mark model for each of the enzyme family. Therefore, it allows domain-based search using Hammer. In 2013, a group in Denmark developed a protein chimer peptide database called PPR, which contains a set of conserved peptides, distinguish each enzyme family from another. The same group developed a search tool called HotPep in 2017 to allow search against PPR for enzyme annotation. In 2018, we developed DBCAN tool to combine the three state-of-art tools for enzyme annotation in one software package for improved annotation of enzymes. However, all these previous tools can only assign a given protein into an existing KZ family. Obviously, this is not enough for people who are interested to learn about the substrate of a enzyme family might target on. There's an urgent issue called polyspecificity in enzyme annotation, meaning that proteins of one family may have different biochemical activities to cleave different glycosidic linkage. For example, GH5 contains proteins with at least 27 EC numbers. These ECs together corresponds to more than 6 different substrates. So knowing a protein belongs to GH5 does not tell you what exact carbohydrate you might degrade. One solution is to classify a family into subfamilies, and each subfamily may contain just one EC number and have distinguishing sequencing features that determine the protein targeting a specific substrate. So this problem becomes a subfamily classification problem, which is not new in bioinformatics field. There are two widely used approaches. The first one is to build phylogenets and then partition sequences based on phylogenetic clustering. Since 2006, using this method, KZ has classified 24 enzyme family into subfamily by manually inspecting the tree topology and their expert curation and classification. There are also automated phylogenetic classification tools, but those were never tested on enzymes. The second approach is chimer-based and does not need sequence alignment, so can be very fast to compute. In addition to PPR plus HotPep that I mentioned earlier, CUP was published in 2019 and eCAMI was published in 2020, both focusing on enzyme subclassification and annotation using chimer idea, but with different clustering algorithms. There are also machine learning-based tools developed for EC number prediction, but not for enzymes. As shown in the oval, our goal is to cluster proteins of one enzyme families into subfamilies shown in the small circles. The colored curves represent experimentally verified enzymes with EC numbers. We implemented a bipartite network algorithm for this purpose. In the network, X nodes represent proteins and Y nodes represent chimer peptides present in the peptide. The chimer peptides are extracted from all the proteins of the family using a sliding window. Some chimer appear in multiple proteins, and some chimer and the chimer protein mapping can be illustrated as a bipartite network. By projecting the network onto proteins, we can get the protein clusters with edges indicating shared chimers and width indicating the number of shared chimers. Similarly, we can project the chimer peptides, which will give us a set of conserved chimers. The detailed algorithm can be found in our paper, but the product of this process will be subfamilies or clusters, and a set of chimer peptides of each subfamily to distinguish one subfamily from another. To evaluate the performance of eCAMI, we used the 24 enzyme families that have already been classified into subfamily through the years by Casey as the ground truth. Most of those families are from PL class and others from GH and AA. Basically, protein of each family have already a subfamily label from Casey. We then ran eCAMI on these proteins to see how well eCAMI clustering results match the Casey subfamily labels. We compared the performance of eCAMI against PPR, HotPep, and CUP. The result shows that in 22 out of the 24 enzyme family, eCAMI has a better performance in terms of the percentage of proteins that can be classified and the classification accuracy. In addition to classification, we also compared the 3 tools performance in prediction, meaning how well we can assign a protein to that corresponding family. We found that in 20 out of the 24 family, eCAMI has a better prediction accuracy in a 5-fold cross-validation experiment. Although these 3 tools were designed for enzyme classification and the prediction, they can be used for general enzyme annotation as well. In order to evaluate their performances on enzyme classification, we extracted all Swiss prot proteins that have EC numbers up to the 4th levels. As we know that EC number or enzyme commission number is a hierarchical classification system with 4 levels. The 4 levels are class, subclass, subclass, and substrate. We focus on the 3rd level, which is subclass. Protein of each subclass all have their substrate level labels, which are used as the ground truth. For example, EC 3.2.1 has 3,438 proteins, and all these proteins belong to 141 EC 4th level groups. We ran eCAMI on this 3,438 proteins, trying to compare the predicted classification with the known EC 4th level classification. We found that for most EC 3rd level groups, eCAMI has a better performance. Here we showed the results for the 20 largest EC 3rd level group, with the largest containing 10,000 proteins and the smallest still contain more than 3,000 proteins. In 18 of the 20 EC families at the 3rd level, eCAMI was able to classify more proteins with a better classification accuracy. In addition to the 3 chimer-based tools, we also included 2 other tools for comparison. E2P2 is a homology-based search tool using BLAST and SciBLAST search. DPEC is a deep learning-based tool which uses 3 layers of deep neural network trained on Swiss prot enzymes. We used 2% of all the Swiss prot enzymes as testing data and the rest as the training for all the 5 tools to see if we can predict the right EC number for the testing proteins at all the 4 levels. As shown here, interestingly, the chimer-based tools all have better EC prediction performance than other tools at all the 4 EC levels in terms of the percentage of proteins that can be annotated and the prediction accuracy. eCAMI consistently has the best performance at all 4 levels. Another advantage of chimer-based method is that they can output the distinguishing chimer peptides for each subfamily in classification and also for each protein in prediction. These peptides represent the key binding motifs or catalytic sites that can be mapped onto a solved or predicted protein 3-dimensional structure In this example, this glycosyltransferase enzyme, the chimer peptides form the activity center, which interact with the sugar substrates shown in green. So to summarize, eCAMI is a new chimer-based enzyme subclassification and annotation tool. eCAMI has the best performance in terms of classification and annotation of enzyme and EC numbers. The chimer-based tools perform better than homology-based tools and deep learning tools for enzyme EC prediction. At last, chimer-based tools have the unique ability to identify the characteristic chimer peptides in the predicted enzymes. So with this, I want to thank you for your time. I will be happy to take questions.